Thanks.
It's just gonna take 10 seconds.
Yeah. Johnny, usually we would like the speaker to make a 40 to 45 minutes talk, followed by 10 to 15 minutes Q&A session.
Okay.
Yeah.
And we are live now.
It's fine.
Hello, Charlie.
Hi, how are you?
Hi, Lulu.
Hi.
Hello, Shijin.
So you didn't go to the SES annual meeting?
No, I didn't.
Okay.
I have class on Tuesday and on Thursday.
Okay, it's a pity. It's almost 10 o'clock. I think we can start.
Okay.
Yeah.
Let's see.
Yeah. Before your talk, I would like to introduce you first. Yeah. Hello, everyone. Today, I'm glad to have Zhang Li to give a talk here. And Dr. Zhang Li Peng is an assistant professor in the Department of Biomedical Engineering at the University of Illinois, Chicago. He got his PhD from the University of California, San Diego, and carried out his postdoc research at MIT. And his research area is computational science and engineering, with a focus on the multi-scale modeling of soft biological materials. And he has co-authored over 50 peer-reviewed journal articles, including 5 published in PNAS. And today he will give us a talk on the physical mechanism of red blood cell splenic filtration. Okay, Zhang Li, it's your turn.
Okay. First, thank you, Xuejin and Zheng Li, give me this opportunity. First, this is my first time to give a kind of a talk, that kind of global talk. So people from everywhere. It's interesting seminar. So all the people in the world around like China, Europe can listen. I think it's very interesting. The second thing is I saw among the organizers so many old friends. So that kind of reminded me the good old days when I was in Boston. So every Friday I take the commuter rail, travel to Brown, and like talk with Xuejin, Zheng Li, Li He, and many others, like joking about our postdoc life and have sometimes very good food together. So that's wonderful. So today I will talk about the physical mechanisms of red blood cell spleen filtration. So this is actually the same title of a recent paper in press. We kind of accepted in PNAS. So first I want to acknowledge, so first is this is really a collaboration work with a French group, especially Annie and Viela and Emmanuel group. And then Alex, actually he is here in the audience. He is the main person conduct a lot of experiments. Really, this French group is really amazing. Without them, it's kind of impossible for me to really understand the process. And Alex is also now a postdoc at Yale University. So besides, also I want to acknowledge, actually I started studying this kind of spleen problem when I was a postdoc at MIT. So Dr. Mindao, my post advisor, and Subra Suresh kind of helped me to trying to start to explore this kind of spleen filtration of red blood cell problem at that time. I'm very grateful, and many other my mentors are listed here. So also the funding support from NSF and American Society of Hematology. Okay. So today my talk will be kind of 6 part. It's a little bit long. So what I will do is actually I will just talk about the physics in the beginning, rather talk about our numerical method. So in the end, hopefully if I have time, I will cover more about the details of a numerical method. So motivation and background. By the way, welcome to interrupt me at any time, so I can answer your question. We can have a good discussion. If you want to interrupt me, please raise your hand or just speak so I can talk. Okay. So motivation. So why I want to study red blood cell pass through the spleen. First, the red blood cell is really it's kind of beautiful. I'm showing here when it is at rest. It has a kind of biconcave shape, although when it's moving, it has kind of bullet-like shape, like cars in a highway. So why it has such beautiful biconcave shape is still not completely clear. For me, it's like a donut, like without a hole, the donut. That reminded me of my life as well in Boston, the Dunkin' Donuts. So why it has such beautiful donut-like shape is not completely clear. I mean, another question people may ask is why the red blood cell designed in such beautiful biconcave shape? I think this question actually is invalid because the red blood cell in the body never are in a biconcave shape unless we are dead, right? If it's not moving, it has a biconcave shape, but even it's moving, never has a biconcave shape. So the only question is what caused the biconcave shape is still not completely clear, although we know it's minimized energy, but more than that, there's a lot of other details. So besides beautiful, the red cell has amazing mechanical strengths, excellent mechanical strength. For example, this is the central question I try to address today is the red blood cell pass through a slit in the spleen, showing here. So it needs to pass through this kind of about 500,000 times during the lifetime. So the red blood cell is 8 micron. So the slit I will show you actually can be narrow as 0.2, 0.3 micron. Then red blood cell can pass without any problem. So in summary, it's why I study red blood cell. It has a beautiful shape, also have excellent strength, very strong. Sometimes my wife thought I was talking about a woman. Okay, it's beautiful and it's very strong. Okay, so that's why I want to study red blood cell. This is another SEN image about red blood cell pass through the slit in the spleen. So here is a very nice sketch. I think Dr. Min Dao probably modified from some older graph by Pierre Buffet. So showing the pathways, not the pathways, how the red cell pass through the spleen. There are open circulation called slow open circulation, closed circulation. Then 10% of the blood need to go to slow open circulation. Then the red cell need to go through this kind of intraendothelial slit, go into the vein, then can go back to the circulation. But if the red blood cell is stuck or go too slow, it may eaten by this MP macrophages, then can cause problems, or sometimes it's a natural selection to remove the abnormal and diseased red blood cells. So another motivation for me to study this is to study the more than 1 billion people suffers from red blood cell disease. Many of the disease actually relate to the spleen filtration. For example, malaria. There are 1 million deaths per year still, most children. So when I was at MIT and Dr. Min Dao and Sura, we helped to study really how the spleen filtrates the malaria-infected red blood cells, and working with a Harvard medical school group. Then sickle cell disease. Interesting thing is original I thought in sickle cell disease, the spleen will kind of stop functional, became kind of dead in the early ages. But recently, obviously, Dr. Mindao and others also get a funding NIH study the spleen filtration in sickle cell disease. I really want to learn more about that. Okay, so the other is like hereditary membrane disorders like spherocytosis, the cell. All these cases, it became rather than beautiful, the red blood cell became ugly and more rigid, so that they have trouble to pass through the spleen. Then will be stuck. For example, in many patients, like in HS, hereditary spherocytosis, rather than have a spleen at the side of a fist, actually it becomes so huge, it kind of blocked, they need to remove. In fact, I go to a science museum, they have this kind of real show of the bodies. I see the spleen actually is even smaller than my fist, right? So, but in patients with this kind of disease, the spleen can be huge. Then the only way to cure it is to remove the spleen. Okay. So the early days, actually, there's some very good paper to understand this kind of spleen filtration structure, for example, by Chen and Weiss, 1970s. In 1970s, there was this kind of very good imaging showing the red blood cell within this kind of sinus or in the spleen, and showing this kind of very nice sketch, understanding how the red blood cell pass through this kind of slit. Okay, then later on, Pierre Buffet and co-workers even measured more systematically what's the dimensions of the slit. For example, as shown in the bottom, the mean length, standard deviation, the range of the length and width is about 1.89 here, showing here. But the most important thing you see, the smallest one, they kind of give us is about 0.25 micrometer. I highlighted using red. I show you later, actually, the red cell, one of main point of our paper is the red cell actually can pass through this slit without any problem, the smallest slit here. So that is the length and width, but on the other direction, you can see these kind of fibers, called the basement membrane in the top left figure. So between these two membranes probably is like 4 to 5 microns, okay? So then one question is, for this kind of 0.25 micron, whether the red blood cell can pass through, or if the spleen needs to open a slit, like showing here, that because in this kind of fibers, they are kind of stress fiber, can contract, can open the slit. But for example, Dr. Mindao and my Dr. Asaro, actually is also kind of my mentor in San Diego, applied the final element to simulate these cases when it's open, what happens. But so in our experiments, especially with the French group, Alex, we try to really test how small the slit the red blood cell can pass through. For example, Viella and Annie and Emmanuel's group, they developed this technique to can actually manufacture the slit down to very small slit size, like W is the width, can down to 0.2, and then LD, the length, D is the kind of depth, we keep like 5 micron, and similar to the spleen. They made this kind of device, then and is PDMS is kind of rigid, so you want to see how small the slit the red blood cell can really pass. Amazingly, we find out the red blood cell actually can pass, like for example, 0.28 micron slit without any problem. That's much smaller than we thought. What I'm here showing a red blood cell pass through these kind of slits, and then in figure B showing actually is a characterization of the actual spleen dimension is 0.28 micron. So, for example, we also did a simulation. So, for example, we simulate the figure showing here is going to give two kind of information. First one, the two cases, one we call dumbbell bell shape, the other is called a tip shape. For these two cases, the top row is the experiment, the bottom row is the simulation. So you can see two things. The first thing is the number highlighted here is the microsecond, is at different time location. You can see the transient dynamics actually can capture well using the simulation by comparing with the experiment. And also the shape actually is not that bad for both cases. I will explain later on which can cause the downward shape and which can cause the tip shape later on. So I think that this is kind of the first time we actually have very systematic comparison between between the simulation and experiments. In the past, even including myself, did a lot of simulation, but it's not really validated or compared with experiment. So this is the first time. Any question so far? No? Okay. So an overview of the modeling approaches, we actually, interesting thing is we're using two kind of approach. The first one, we used a in-house final element code and coupled with a boundary integral. The final element is for the cell membrane, then boundary integral is for the fluid, for the transient dynamics, like including the fluid structure interaction. This is really simulating the case when the cell can pass through the slit relatively easily. Then the second approach using abacus explicit without considering the fluid. In this case, we study the retention, which means what's the critical force to push the cell through. Why we need two approach? In a simple way to explain is that because imagine if the cell became harder to pass through, the transit time for it to pass through may take it go to approach infinity. Became longer, then it became impossible to simulate the fluid structure interdynamics with such long time, right? So we don't need to care. In that case, it became quasi-static. We do not care about the real dynamics. We just care about, okay, it can pass or not. And other reasons, including, for example, in this kind of retention simulation to predict the critical force, you really want to use in the actual stiffness of the bilayer, which is huge. Is kind of several order higher than shear modulus. If using that, actually will lead to very small time step size. So it's even make the fluid structure interaction simulation impossible. But no matter which method we are using, we actually, for the constitutive law of the membrane, we use a new lattice model which considers the distribution of the spectrum length orientation and even spectrum unfolding implemented either in the in-house final element code or the VUMAT for the abacus user subroutine. So I want to show some example here. For example, the cell pass through a slit. This is a really slow one. The next one, then this you have for example, tip. Sometimes can from this kind of fingertip in front. It takes a very long time for us to figure out why it's from this kind of fingertip in during the process. So Alex and others did a lot of experiments, for example, summarized in the two tables here. For example, with various dimensions, for example, it can be large as 1 micrometer, then down to 0.28. And more importantly, we did for two different temperature, at least. One is at the body temperature, 37 Celsius. Then others is like room temperature, for example, 21 degree. Amazing thing is, you see, for all this kind of at the body temperature, even when the slit is smaller than 0.28, there's no retention at all. The rubber cell can pass through such small slit, but when at room temperature, even the slit is even larger, 0.43, you can have significant retention. So basically what I'm saying is here to summarize is the rubber cell actually can pass such small slit at 0.28 micron because the cell is about 8 to 10 micrometer. But this can only happen in body temperature, but when at room temperature, then have difficulty to pass. I will explain why later on. So first thing I talk about is the geometric aspect of this problem. So in 2016, we work with Igor Tolkien and Darmin Dalsubra. We published a paper, PS paper. So trying to explain this kind of observed surface area volume relationship of a red blood cell population. You can see here, Kenhan and Burden in 1968 published this very famous paper, although it's not famous, but it's really famous. So they have this data here. Then later on, somebody did a more refinement, like a Gifford, like a red point here, showing the relation. At that point, our observation is, you see, the red blood cell, the surface area volume here, showing almost this kind of a linear relationship between them. But you imagine if you just scale up and down of a biconcave shape, like increase the, just keep the same geometry, scale it down, scale it up, then we know the surface area is increased of length square, right? Volume is increased length cube, right? Then the surface area volume should not follow a linear relationship, but it's more like a nonlinear fashion, like probably power 2/3, something like that. But why is this? At that time, we kind of did some analytical calculation, then show, oh, if this red cell pass such geometry shown in the bottom, and it might kind of cut off this line as showing the dashed blue line, the analytical one. So, but this is an alternative explanation, because in the early days, even in the 1968 paper by Kenham and Burden, they have a theory, instead of saying spleen, they say if the cell passes through a capillary, showing on the left as such small, actually it can also kind of bound it or lower bound. Basically, for this solid line, any cell above the solid line will be able to pass, and below the solid line will not be able to pass. That explains kind of the cutoff of this kind of surface area volume ratio. Okay. This is an amazing paper, although very old. I saw, oh, who is Kenhan and Burden? Realized actually Burton was a kind of pioneer doing the micropipette aspiration of red blood cell. I thought Kenhan was his student or postdoc, but turns out really, I realized Kenhan was kind of the president of American Society of Physiology. So he's a very kind of famous guy. This paper I will show you later on has a lot of good insights. So another insight from this paper is showing there's a difference between normal people and the people with the spleen removed. I showed you the picture when the spleen became enlarged, then it's removed. You can see the kind of distribution shift significantly. So, which means the spleen actually might play a major role to defining this kind of surface area volume relationship. Then in our recent work, we realized we tried a lot. We realized something might be more interesting thing is this. For example, imagine if the red blood cell, the surface area is large, allow it to deform it to a two sphere connect with a tether, okay? Then this configuration in principle can pass through any pore, no matter how small the size is. Why? Because you see, this is the infinite tether. You can always insert this infinite small tether into no matter what small the pore is, insert to it, then grow this sphere right on the other side. So which means it can pass through any pore, okay? So this is interesting. Then we realize in experiments, you can measure it's indeed, although our slit is not really exactly the same as the left, as a kind of inclined wall, but it's indeed show two spheres, as some limit case, two spheres both sides. But we don't know whether it form a neck between them or not. To do that in our simulation, showed in the bottom left, it showed in some cases, indeed, it formed this kind of very small neck tether connect these two. And more, we did also the 3D confocal imaging, and it shows that, yeah, then in experiments, sometimes indeed can form a kind of neck between them. In fact, then if you need two equal sphere model, it's very easy to calculate the surface area volume ratio if you get two sphere, right? So that you can just establish the volume to area very easily. Then you can actually bound it by this green line. Okay, so it's cut a little more. Then in our case, we had another case, the cone, because as I said in our experiment, it's not exactly a slit, but I have an inclined wall. So the both left and right side should be a sphere plus a cone. So this kind of simple two equal sphere model can explain the half very clearly. That's amazing. But actually, you know what? At that time, we thought our idea is so exciting and interesting, but it's not. Turns out it's not new at all. In 1968 paper I showing here, for example, they said the possibility that river set pass through this kind of small slit can be analyzed. The optimum case to conserve area would be as what remain a segment of sphere on each side, then kind of divide on each side, although they didn't say it's connected by a tether. This form of diapetes of red cell is theoretical possible, but on pass through any hole, regardless of how small it is. So you can calculate this easily. That's what the curve I showed. But they said, however, the calculation is obviously unrealistic since it ignores any rigidity of the membrane or even bending rigidity. Okay, so there's basically this. Although they kind of thinking about this idea is fantastic, but they thought maybe it's kind of not really the case. But we think it is. So then that's why I want to show this slide. The thing is, you see, the things ignore any rigidity of the membrane and the acute curvature is involved. So we need to study that. So that leads to the mechanical aspect of the problem. So far, do you have any questions about what I mentioned? No? Okay. So the mechanical aspect, as I showed you, one important observation of the experiment is the cell can pass the very small slit at body temperature, but not at room temperature, has significant retention, like 16 or 23% retention. So we try to understand why. Oh, somebody drew something. Lin Jiehe, you drew a curve. So do you have any question on my screen? Okay. Maybe okay. Maybe it's a mistake. So we actually using objects explicit, we consider to study room temperature versus body temperature. What is the delta PC is a critical force for its pass. You can see actually the room temperature, the critical force is much higher than the blue line, which is the room temperature critical pressure, is higher than the solid the black line at the body temperature. Then if we think about the physiological pressure is 500, is actually the sphericity for its pass, it will be much smaller in the room temperature. So what happens? What's the temperature effect on the cell? So at least we showed there's three effects. First, one thing I will show you later is actually at body temperature is decreasing the unfolding force, F half. What is F half? F half is the force to unfold 50% of the repeat in the spectrum network. Okay. So it had been established by experiment in the past. This value F half, which means the difficulty to unfold the spectrum repeat, can be smaller in the body temperature. Then second thing is, of course, everything kind of expand at higher temperature. So there's increased surface area at body temperature. Then also people measured the area modules of bilayer decreased a little bit as well. So we actually studied different effects. For example, a combination. For example, people showed very likely the force to unfold the spectrum 50%. It's about only like one order lower, 1 piconewton at the body temperature rather than 10 piconewton at the room temperature. Also, the area modulus decreased a little bit at the body temperature, but the surface area also increased a little bit. But we find out among these three effects, the first one is the dominant effect. It have a biggest impact on the critical force. This then followed by the second reason, then the third one. Okay. So then we did a more detailed study. For example, if we in the figure A, if we increase the at room temperature, increase the half to a very high value from 10 to 100, it didn't change that much of the curve for the cell to pass. But at body temperature, if you increase 100, actually, you see, my point is that if you look at the right figure, at F equals 1 piconewton, then most of the cell can pass. But if F equals 100 piconewton, you can cut a significant amount of the cell. What I'm saying, of all this curve, the cell below the curve will not be able to pass, but above the cell will be able to pass. So here is more data showing. In fact, we also, as a way, I will not go to details, from the experimental dynamic data, we can kind of try to estimate what's the critical pressure for the cell to pass through. For example, for the body temperature, the black dot is the data from the experiment. Actually, that matches relatively well with our simulation data as well, although there's no very difficult way to directly measure the critical pressure. Is there any question?
Oh, yes.
Yes.
Hi, Zhang Li. I'm Guanshen from Brown.
Yes.
I have a question.
Yes.
What's this line for 500 pressure drop?
Oh yeah. So we kind of think about it as the physiological pressure in the spleen. As I referred to you, there is the paper. Actually, they kind of measured the pressure in a real spleen. So that's our estimation of the physiological pressure drop between the sinus through the spleen slit.
Oh, this pressure drop in the spleen.
Yeah.
This pressure drop in spleen is close to near the slit or just the spleen?
Yeah, that's a good question. I think they measure is the whole spleen. Whole spleen, they measure probably like 1,000 to 2,000 Pascal, but 500 mostly is in the slit. Yeah, because they have a wide range. Yeah.
Oh, so you mean this value is the arterial and veins, yes, in the spleen?
Yeah, the artery vein probably is 1,000 to 2,000. Yeah, this what we say here is probably across the slit. Yeah, estimation.
Okay, another question is I saw the pressure drop is so large than the previous paper, such as PNAS 2016. In 2016, the pressure gradient I think is about one part per micrometer. Yes.
Yeah.
So at here the pressure drop. This is a pressure drop. Yes. No pressure gradient. This pressure drop is so large. Do you when you did experiment, do you find some? Do you think it can cause some damage to the cell when cell pass through the slit?
Oh yeah, that's a good question. I don't. Yeah. For example, I'm not sure if we see here. That's a very good question. You can see. Typically, that's the pressure. The third column here is the in-slit pressure across the slit. That's we kind of using, for example, you can see up to 1,500. So it didn't cause any damage. Even 160, 23. We for normal cell, we didn't see any really damage. Yeah.
The cell just pass through the slit once, or you use the one cell to pass pass the slit many times?
Oh, that's another very good question. In the current paper, we only passed once, but actually I wrote another paper along with another group. We published many times, but seems there is that's another aspect of the mechanical kind of cyclic say, for example, you pass this one, for example, 200 times, then that can cause some problems as well, but once it's not. So in this paper we let cell pass the slit many times, the same cell, then it is indeed has a impact on its mechanical properties. Yes.
What's the width in this paper, the width of the slit?
The width of slit, I think that's a good question, because for example, when we look at it here, that's actually, maybe we can directly go through the paper if you want to see. I think I was looking. It's 0.85, 2 or 3. You can see here. So it's 0.85.
So the slit, the width of the slit is less than the current paper. Yes.
It's larger. The current paper is but no, I cannot say that, because in the current paper we have a lot of different slits, right? So from 1 to 0.28, but I think the typical one we used probably is this kind of one, like 0.4. But this is indeed a good question, because I think you refer to our old paper on that spleen pressure, because we got that at that time. We don't know what is the pressure in the spleen. We kind of estimate from Y.C. Feng's book about the pressure drop in the capillary, but I think that estimation is too low. So that's why later on we actually find a paper that actually explicitly measured the spleen slit pressure drop, but I cannot immediately figure out which one it is. But I can show you later. Probably I can.
Okay. Thank you. And another question is, can you change to your slider?
What's your question? Sorry.
Can you show your slider?
Oh, slides? Okay. Yes.
Because in the spleen, the intracellular cell, the geometry of the intracellular cell is almost silent. Yes.
No. The slit is something like this, shown here, for example, something like this. The slit. It's not a cylinder.
Okay. In the experiment, I saw the geometry.
Oh, it's the slit, like here.
Yes.
W is much smaller than L and D.
Oh, the L. I mean the L. The L. What's the length? What's the length of this? What's the value of L?
Oh yeah, that's a good question. For example, in experiment, it's measured about like 0.9 to 3. This is a real spleen. Then
this is the diameter. Yes.
No, this is the length. You see in the paper, it's the mean length is this one. Then this is the width. Then in our experiments, the length is roughly like 2, or sometimes can be 1, like 1.4 to 2.5. It's consistent with the in vivo measurement of the spleen slit. The L is this one. If you think about the arrow here, is the length of this black arrow. Then it's not a cylinder, it's a slit, like showing here, showing in the top graph here. It's kind of this slit.
Okay, thank you.
Okay, that's really good questions. Then, for example, maybe I should go on. It's out of the time. So we actually, for example, in this figure, we're showing when the cell pass through, actually the gray area showing here, where is the region the spectrum will unfold it. For example, this is the pre-post stretch. You know, how many times the spectrum was stretched. For example, if our estimation is if you stretch about like 3.9 times, we'll probably unfold the spectrum. I can show you the model later on. Okay. Then another possibility we say is I talked about the temperature effect on these three things. One, you may argue, because the red blood cell has the piezo-1 channel on the membrane, as I've shown, will pass through such small slit will trigger activate the piezo-1 channel, then lead to the volume decrease, make it easy to pass through. That's a obvious question to ask, right? The natural question. So that we study the piezo-1. So to study that, actually put like a solution with and without calcium, because piezo-1 supposedly is allow the calcium get into the cell, then activate Gado's channel, then cause potassium leakage and water decrease, right? But our result shows no matter is calcium outside and not, actually the calcium signal is very similar. So interestingly, there is a calcium signal there, even external fluid has no calcium, which means there may be some calcium storage bind to the protein, then kind of released. It's not related to piezo-1. So for example, in this particular diagram, we showed that, sorry, let me hide the meeting panel. So for example, the white is the upstream calcium intensity, downstream is the gray one. So it's always the same, no matter there's calcium outside or not. Also, we did a lot of different pressure, even although we didn't add in the paper, we even used the like a piezo-1 inhibitor, Gado's inhibitor. It doesn't make any difference. So our conclusion basically is the piezo-1 is not really activated during this process for some reason. Maybe because the tension is not high enough, or because the people showed maybe there's only like 20 copies of piezo-1 on red blood cell. It may be a little bit too small. Regarding piezo-1, our story here basically is a negative story, saying piezo doesn't work. But for a side story, I want to show another one is the leukocyte neutrophil need to pass through the spleen slit, not spleen slit, sorry about that, pass the endothelial slit from the blood vessel in the top part to destroy the bacteria in the tissues. They need to do this as well. But recently, we wrote a paper. Actually, in this paper, very interesting is we did all three. We did an in vivo study of the neutrophil pass through a pore slit in the mice. Then we did a simulations in silicon of the very similar approach, similar to the neutrophil. PMN is a neutrophil. Then we also did microfluidics to see, but if fluid is much larger, the pore, for example, we showed this actually can activate piezo-1 in this process. Then we estimate the membrane tension on the neutrophil and related to the activation probability of piezo-1. And the more interesting thing that the main point of this paper is actually if it's activated, for example, if you let the neutrophil pass the pore smaller than 5 micron, actually it's increases the bacteria killing capability by a lot. So which means when the cell passes through this kind of slit, maybe activate some mechanism, make it more capable to kill the infection. So this paper was actually just accepted in Immunity. That's probably the journal have the highest impact factor I ever published, 43. But the interesting, weird thing is I never heard this of journal before. Maybe most of you didn't heard of this journal before either. But it's a very interesting work on the piezo-1. It's a positive story rather than the negative story in the red blood cell side. So next we'll talk about the transition dynamics a little bit. So when I say transient dynamics, which means how fast the red blood cell pass through. For example, y-axis is 1/TT. What is TT? TT is the transient time, like how much time take the cell pass through a slit. 1/is inverse, right? So in this diagram, we showed a systematic comparison between the experimental data, which is the solid dot and the lines, with for various dimensions of the slit, and the comparison with the simulation modeling using finite element with boundary integral. You can see they really match well. We are a little bit surprised. It's good, so such good comparison for different pressure. So, for example, this is really a mean transient time because the red blood cell has a population. For example, this is a typical distribution in an experiment for many cells, how the cells, some are outliers, maybe some, but we get the mean value. So here, for example, there are many other data, for example, different slit between experiment and modeling. I will skip some of them, and sometimes it's even for different temperature. And more, when we submit this paper, also the reviewer was asking, oh, can you do a little bit more, for example, controlling the membrane rigidity, for example, by adding dynamite? So we did. Actually, we added the dynamite. Dynamite is known to increase the stiffness, but interesting thing, we realized that, I will not go to details, is we find that the transient time indeed is slightly increased when you add a dynamite, but we find the simulation shows it's not because it's increased the rigidity, but actually it's increased the membrane viscosity. I will not go to detail, but actually that's a very good suggestion from the reviewer, so we just that. And in generally, when you put all the data together, we find that the transition time, which this S, what is SI, is the slope of the transition time shown here, actually change with like power 3 with the width, W is the width. Okay, that's in our paper we explained in a simple way that it's mostly the flow past the slit is like a 2D positive flow. But actually, we did more. For example, with different osmotic pressure, under different osmotic pressure, the cell volume will change. We also study its effect, for example, experimentally and modeling to show actually how the it's very interesting. If you change the osmolality, there's a two kind of opposite effect. On one side, if you change the, for example, if you increase the osmolality, the cell volume will decrease. When the cell volume decrease, of course, purely from in terms of volume, the cell should pass faster, right? Because they have less volume passed. But on the other hand, if you decrease the volume, the concentration of the hemoglobin, MCHC, inside. But then the viscosity of the hemoglobin will increase dramatically because it's a highly nonlinear fashion of how the viscosity of the hemoglobin change with MCHC. So the net effect we find out is still slowing down a lot, even the volume is less, okay? Or paper in physical fluid, we did more systematic analytical theory on how the cell passes through slit. We did analytically, we derived even analytical equations. So how the transient and how fast the cell passes through the slit, how can it change with the viscosity inside, the width of the slit, and the depth, etc. We published in with two amazing undergraduate students in my group. We developed a theory, published one in physical fluid, and another one reviewing now in micro machines. So the next question, for example, this is the one will be stacked. Okay, the cell, I show you more videos. Then there's another one. This is more like a sickle cell pass through. So why I'm showing this video? Because I will go back to the question, the deformation shape. This is a boundary integral versus coupled with finite element showing the cell pass through this slit. So the boundary integral was using the Stokes flow. So you can see actually it shows very well matching the transient time, but also the shape. Here, for example, as I mentioned in the beginning, for a very long time, we actually don't understand why some cells showing this kind of dumbbell bell shape, but some cells from the slit. But then using our boundary integral simulation, for example, if you keep the same volume, but increase its surface area, for example, then you see, gradually it starts from this tip. So turns out it's not that difficult to understand. It's just the volume is or surface area is larger or volume is less in this kind of tip information cell. So that explains why in our experiment, the tipping happens a lot for sickle cell. Because sickle cell is dehydrated, that has less volume. So that's the tipping happens. So finally, I think I have a few minutes. I will go through a little bit the computational approach we developed. So for the membrane, we're using a more latest microstructure-based model. What is the update for this model? Because people develop a lot of kind of Weber cell membrane models, like showing here is the structure of the Weber cell membrane with a spectral network. So, but one thing we recently did is traditionally people believe there are 33,000 junctional complex, about like 0.2 million spectrum monomers in Weber cell. That's the belief. But recent data, for example, here shows actually the spectrum can be 3 times more than we thought. Okay. The spectrum is 3 times more than we previously saw. Then how that changed the picture of that? So instead of 33,000, we may have like 100,000 junctional complex. So that's the first new data we got from the protein omics data. Okay. The second one is actually from the cryo-EM data. Actually, we can get some like a distribution of the spectrum tetramer. You can see, typically people think the spectrum tetramer length is about 70, but this data showing it has distribution, but even the mean value maybe it's around 50 or 40 is like a less. That actually is consistent with the protein omic data, because basically saying both data suggesting the spectrum network is more denser than we thought. Okay, so based on this one, we trying to incorporate these two new information to the model and trying to validate it with the micropipette experiment. So I will probably skip the details. We derived this model based on new, but an innovation thing we did is we can see the different distribution of orientation, also the distribution of the initial length we got from the data. Then this is the more fit with the experimental data by Richard Waugh. We worked in this paper together. He did the pioneer work on the microbiome, trying to understand what really happens. So that's the microstructure. Although there's a lot of other insight here, I will not go to, but basically that's kind of the major part of the microstructure-based model. Another important thing is we incorporate in the unfolding of the spectrum. For example, this is a total length, including a number of folded spectrum domain and unfolded one. So the total length will be this phi. The phi is how many is unfolded. So it depends on the half. That's the number I mentioned. The force to unfolding 50%, then including this in our model. Okay, so we did it recently for another purpose. For example, using similar things for a laminin filament in the nuclear envelope. We did the all-atom numerical simulation, all-atom molecular dynamics of the spectrum. Both spectrum and the laminin then fit this kind of unfolding model. It's kind of under review. Then I also probably skip this one. This is a side story. We apply a similar model to study the nuclear laminin at the spectrum network of the red blood cell by including the orientations, the density from the cryo-EMs. So why we studied that? Because when cancer cells pass through the slit, the nucleus was squeezed a lot, like the red blood cell. So people study how the nuclear envelope may rupture when they squeeze through a microfluid device. Then we did a lot of simulation. For example, we even explicitly modeled the chromatin inside to understand on one side how the different components of the nucleus contribute to this mechanical property of the nucleus, including chromatin and the nuclear laminin. On the other side, we study if the deformation, how it's like maybe change the conformation of the chromosome. So the final part is the boundary integral formulation. Actually, I did this here, 2019 paper about the method. So it's a little bit very difficult because in boundary integral simulation, you have the Newman boundary, the Dirichlet boundary. You actually form three equations. For example, the red circle showing here is the unknowns. You have three unknowns. For each equation, you have three equations. You kind of solve this one. But unfortunately, it leads this to a very complicated code of thousands of lines. So that's how my student struggles. So that's why I prefer my current student not to try to do something like this. It's too tedious. So I think that probably is all I want to tell you. Maybe I will stop here rather than showing more slides. Then I will be happy to answer any questions or just any comments. Yeah.
Thanks, Zhang Li. You did a very solid work on multi-scale modeling of a biological cell from just you mentioned this from 3 different levels. Yeah, and it's very good talk.
Thank you.
Now it's time for questions. So if you have any question, just mute yourself.
Yes, I think a question for Xin.
Yeah, very nice talk. I have 2 questions. The first is what's the effect of the wall to membrane interactions? Did you treat any chemicals at the wall?
Yes. Yes.
With the slit?
Yes, they treated to make it more less adhesive. So make sure the wall is not the cell is not adhered to the wall. Yes.
I see. The second question is so after the cells passing through the constriction, they relax back, right?
Yes.
Is this relaxation mainly due to the membrane tension or the viscoelasticity of the cytoplasm? And is there a way to distinguish the two just by looking at the relaxation?
I think these two in for red blood cell is kind of the same. Is because basically it's the viscoelasticity or mainly the elasticity of the cell membrane. There's nothing inside the cytosol because it's just a bag of the hemoglobin solution, unlike other cells. When you say tension, it's a little bit tricky here. It's part of the elasticity of the red blood cell membrane. Yeah.
Okay, thank you.
Yeah.
Other questions?
Hi, Zhang Li.
Yes.
This is Shu Hao from Professor Shijini's group. I have 2 questions. The first one is that how do you measure the F-half in your slides? It's from the experimental data or simulation?
Oh, it's kind of from experiment. There are several paper. For example, let me figure out where the especially from Dennis Dirichlet group, they're using the AFM or optical tweezer to stretch in a single spectrum. Then they kind of measured what is F-half. Let me see if I cannot find it. For example, this one. if you look at this one, path
Okay.
shift and thermal softening, for example, we talk about the temperature effect in temperature and coupled forced unfolding of spectrum domain. They try to measure this. And in this one as well, this is my colleague, turns out, James Lee. And they also measure this one. And this is another theoretical work by my doctor advisor. This is purely theoretical, but these two are experiment. And more importantly, there is another, maybe it's not decided here. Actually, they measured also different the unfolding force of the spectrum, even for different domains. Yeah. So, but I think to adjust your question is, roughly we know the F half at maybe body at room temperature, but we know at body temperature is lower, but how much lower is really one. That's probably a reasonable guess, but it's not the exact measurement. We know it's lower. Yeah. For example, you can read the appendix part of this one. The last few paragraph of this paper is discussed this one. Yeah, the difference between and also this one, the temperature effect. Yeah.
Okay, thank you. And another question is that I see that you discuss the dynamics of the red blood cell when transit the slit, and the membrane viscosity and the cytosol viscosity both influence the dynamics.
Yes.
Which one is more important in this process?
Obviously, the cytosol is the hemoglobin one is more important. Yeah. I think this maybe raise a question is, that's a very good question because, in the original paper by Erwin Evans and Richard Hua, when they measure the membrane viscosity of red blood cell, they're using micropipette aspiration. Then they claim is the membrane viscosity maybe is more important than the cytosol. Then the argument is the shear rate in that micropipette experiment is similar for the cell membrane and the cytosol. I think that is not true because if you think about the micropipette aspiration of the red blood cell, the shear rate in the cytosol is in I don't know how is the R direction, but the shear of the membrane is in a different direction as well. So they cannot be compared. So in that aspect, I think the cytosol, which in this case the hemoglobin solution, is obviously the more important one. Yeah, because we even showed that the how fast it's transcended is really depends on the hemoglobin solution. Yeah.
Okay, thank you.
Yeah, the other one I show here is this in this paper, the conformation stability. They also did a measurement of the unfolding of the spectrums for different temperature as well. This one is an experimental work as well. Yeah, probably this one as well. There's a lot of measurement. Yeah.
Yeah, thanks.
Other question?
Okay, Professor Peng, I'm So Wang from Xuejin's group too, and I also have two questions. The first one is in the temperature effect part. How do you model the temperature change in your simulation? I'm curious. Did you change the mechanical property based on experimental observation or some more specific chemical interaction?
Oh, that's a good question. So I list the three here. For example, I think your formal lab mate your lab mate asked the same question. This decrease in this one, right? As said, is kind of measured in the several papers. Although I said it's not like the 1 piconewton per micron, it's not the exact number, but at least they estimate the this one is much lower at body temperature. Then increase the surface area is easy to understand because you increase to 20 degrees to 37 degree. We know the area expansion coefficient from which one you can directly calculate what's the area expansion. Then this one is also, for example, changed. This is both are experimental measurement from different temperatures. This one, actually, I don't have. If it's easy, so thermodynamics membrane. What about cell membrane? For example, if you go there, is this one. So in this paper, they measured both the thermal area modulus, how they change with temperature, and what's the thermal expansion coefficient as well. For example, here is the thermal expansion coefficient, so you can estimate what's the area expansion. Then also showing the how the bilayer area modulus changes with temperature. Yeah. Does this answer your question? For example, this one is this one. Then at body temperature, at room temperature is about 450. Yeah.
Yeah. Okay.
Then yeah.
Yeah. Thank you.
Regarding the other question, I think Guangshen mentioned is here. So maybe I found it. For example, maybe the table or not. Maybe not this one. For example, maybe it's this one or not. I believe that they have a table somewhere. Maybe I misunderstood this paper. Yeah, sorry. Maybe it's a different paper. Sorry about that. Or maybe this is transient time paper rather than the pressure paper. Yeah, this is early measurement by McDonald of the transient time. Yeah, this is a different paper. My bad. Yeah, I was trying to figure out the paper on the pressure in the. Oh, I think it's this one. I found it, but I'm not sure it's in my system or not. Yeah, this one, they measure the intra I think you also, yeah, in your paper also cited this one as well. So maybe you already know, right? Because this paper, they measured the intra-spleen pressure and index of this one. We should look at this one. Yeah. I don't have this on hand. Yeah. And any other questions? Or?
Yeah,
Yeah,
Johnny, I think sure. The last
another
one.
question. Yeah.
Go ahead.
You mentioned that the width of the slit is very small. It's 0.28. But as far as we know, when we want to make a very narrow microfluidic chips, so we found it's very likely to collapse. So I'm wondering, how can you make a so narrow slit? And what's the material of the microfluidics? Okay,
good question. Let me show a little bit here. Is from speaking, I'm not the right person to answer the question because I didn't do the experiment. Alex, he did it. Do you want to talk, or maybe I just show some, because in the appendix of our paper is
I can explain if you wanted to.
Yeah, for example, the method itself is mainly published in this paper. If you want to look at it, this one small paper several years ago, but in our appendix, it's also explained. Maybe Alex, do you want to add some?
Yeah, so it's a pretty long process with a different step, but what is very important is the fact that usually in the microfluidic field, let's say, people are doing those kind of channel by using optical lithography techniques, and this is kind of difficult if you're just simply using this optical lithography technique, because you cannot go below 2 micron in width. So with this optical lithography technique, we also add an electronic lithography technique and the chemical etching to create a mold, which is very, very thin. And then using the commonly used PDMS techniques, we use PDMS to create those chips. So the material for the experiment is PDMS coated with a BSA in order to do not have any interactions between the red blood cells and the PDMS itself. And how we create this is by using a lot of techniques from optical, electronical, and chemical etching.
Thank you. So I found the paper for Guangsen. If let's say, for example, here is so you can see here, this is different between different mean inch pressure. If you convert this from for example, let's say 20 milli. So you see, if 20 milli mercury is about 2000s. So here is about the range in the spleen they measured. Yeah. Guangsen, does this make sense to you?
Question left.
Sorry?
Question left.
Oh, Guangsen left. Oh, sorry. Okay.
Yeah. Yeah, it's 11:10. I think we can stop here today.
Okay. Thank you.
Yeah, thanks Zhang Li for the excellent talk, and thanks for all attendance who joined us in today's webinar. Yeah.
Thank you. Thank you again for your invitation. Yeah.
Yeah. Thanks, Zhang Li.
Thank you. Thank you, Alex. Yeah.
Mhm.